So, welcome to the next video in our course B01 on data management. So here we're going to talk about data collection, data catalogs, and recording data. So we know that we have need appropriate methods for accurate data collection, and that we should use metadata standards that are relevant for our community and use existing data conventions for consistency across our data sets to link them with other existing data sets. So we know, for example, that if you take an image, a photograph, or record an image, that you may do certain things to make it more clean or make the fluorescence or whatever we're trying to show on it to be more clear. So that's the difference between our raw data, which is the original image, and then the process data. So raw data might be something you share in a primary database, and the process data might be something that we share in a paper or whatever. And what's important is that we have metadata linked to that, and that metadata lets us know if that image is relevant for other researchers, for other studies, is relevant for comparison, maybe in a few years' time when you're trying to review previous work. To record all this appropriately, we have to think about how we record it ourselves. So lab notebooks are a starting point for that. Hopefully we all use digital versions, and modern digital lab notebooks let us record text or images. They're searchable and taggable. They're video and audio, code and website links. They're shareable. They have templates, and you should be able to edit them online or offline. And I think at our institute, most people use OneNote, which is typically good for most things. And that's kind of the standard convention in short. That's so we might have a structured or lab notebook such as this. So we might have multiple notebooks, and then inside there we might have particular sections or projects. And each project may have different components inside there that are delimited across different pages that are easily understood by people that we're working with. And they may have subpages and so on, that it's we've got a systematic structured hierarchy that is easily interpreted by someone who's someone different from ourselves. And inside these lab notebooks, what we want to use is a standard nomenclature. So a data catalog is metadata associated with particular data sets that is easily understood. So for example, we got results from a database search here where we're searching for the foot and mouth disease virus on the SRA on the NCBI website. And you see here that we have metadata appearing about what we're actually looking up in terms of our hits on this database. And that metadata is very, very important. That's what should we be trying to get here. So these are kind of data catalogs that reflect our data sets. And this saves time. So you're trying to find particular data sets. Then how do you do that? You go to a database by whatever mechanism and try to obtain them. So data catalogs using standard nomenclature metadata is very, very important. So to achieve this compilation of metadata, what we can create is called a data dictionary. So metadata should describe the contents of the structure of the data sets, include definitions with column headers. So these examples of this further down here. So amount of variables, I might describe their the type of data here. That's binary, numerical, or label, the units where relevant in millimeters, for example, and the ranges of the data set is also useful. And we might include a description as well. So sometimes the labeling or type of data may not be clear, so it's good to include a description of what the data really contains, such as the subspecies of infected viruses refers to the binary categorization, size of the cells, the infection status, and so on. An example of this is here, where we have a series of these are the columns headers at the start, and some of these are a bit opaque. For example, sac date may not be obvious to your eye, and this is the date of the sac, but again, that's not very clear. That's the date when the meat was sacrificed in the description. So that's helpful to understand what actually really is contained inside there. In the same way, crumblers actually refers to if the mouse stored food in their bedding. So these descriptions are important so we can understand better what they actually mean. So wherever possible in data dictionary, we want to use standard nomenclature that's easily understood. So be simple, be clear, be consistent, not sesquipedalian. So lanes basically create things just to be different. That's not necessary. An example of this is microbiome research, where people's tendency to use different terms such as stool or feces or other terms to refer to the same thing mean that if you're just trying to search different databases for these things, you'll find this it's not always straightforward to achieve because things are inconsistent. So again, going back to being simple may mean using the same term over and over again, but at least it's consistent. It's more easily indexed and more easily searchable. There are there's lots of information on data standards depending on your area. For example, for metabolomics data, there's the Metabolomics Standards Initiative. For proteomics data, there's minimum information about proteomics experiments, and so on. There's established ways of having minimum information, so that's minimum metadata linked to an experiment. An example of this is what happened during the start of the pandemic, that a lot of scientists moved into the area of SARS-CoV-2, but they lacked experience in linking virus genome sequences to data management. And as a consequence, at the start, in the first, from the start of 2020 up to May 4th, when this publication counted, the host was not listed in about 46.5% of the biosample submissions. So there's a real lack of metadata for these particular example. Similarly, if we look at the number of metagenomes in the SRA, fewer than a third actually have environment metadata linked to them. So for many experiments, therefore, the majority will just not be useful. If we look at viral metagenomes, only 23% had their locations specified. So this kind of is just these are just a few examples to really, really illustrate that if there's not good metadata linked to samples, even the known absences of information, that kind of limits what we can do with them in the future. And what's noting is that this minimum information approach is the lowest barrier. It's not trying to achieve a new world record. It's a very simple barrier that's just aimed for basic data submission. And if you do this at the start, this will be easy to achieve in your project. But if you do it at the end, it will mean that you actually spent more time than you should have otherwise done on trying to manage data standards within your project. So that concludes this video.